you all for coming. Thanks for that kind introduction. I'm Will Hudson. I'm at Baylor College of Medicine. I've had my lab here for about three and a half years, and I'm really excited to share some of the work that's come out in the early part of our lab. So I'm going to have kind of three. I can hide these controls. Sorry. All right. The importance of CD8 T cells in immunity against cancer and viral infection. I'm going to talk then about two stories that we're working on in the lab. The first is the function of this molecule called CD7, as well as some new tools we developed for looking at T cell fate within the tumor microenvironment. All right. So CD8 T cells, I think a lot of you are familiar with what CD8 T cells do, but I'll give you a brief introduction in case you are not. All nucleated cells in mammals express MHC class 1. The purpose of this molecule of MHC class 1 is to present peptides that are generated within the cell on the cell surface so that they can be recognized by this class of cells called CD8 T cells. CD8 T cells and all T cells express a T cell receptor or TCR. And when they recognize their antigen through this TCR peptide MHC interaction, CD8 T cells will hopefully kill that cell in order to eliminate the pathogen within it. They'll also produce cytokines in order to amplify the immune response, and they'll also proliferate, generating more of the same CD8 T cells with the same TCR in order to recognize the same antigen. This is very important. We've known this for a long time. This is very important in controlling intracellular bacteria and viral infections. And really, in the past 2 decades, it's become very clear this is also extremely important for anti-cancer immunity, and it's really revolutionized the treatment of cancer by targeting CD8 T cells that are tumor specific. So the TCR is going to play an important role in what I'm talking about today, especially the latter half of my talk. And so it's this T cell receptor that's responsible for recognizing antigen, for recognizing peptide MHC complexes. This is actually generated by a pretty complicated mechanism of DNA recombination, which can generate somewhere on the order of 10 to the 13 possible TCR sequences. Of course, the diversity is not that true in actuality, but you don't have that many cells, but these are very diverse sequences that's created during thymic maturation. And importantly, this TCR sequence doesn't change throughout T cell differentiation. Unlike B cells, which can edit their B cell receptor, TCRs remain fixed throughout a T cell's life and fixed throughout all its progeny, which makes it really useful for tracking T cell fate over time and space. So how do CD8 T cells exude their function? And well, the main signal is that TCR stimulation. And so you have naive T cells, which are generally surveilling. They can move around a little bit in the blood and the lymph nodes. They're quiescent. But when they encounter antigen, such as an infection or a tumor, they will differentiate into effector cells. These cells are cytotoxic, meaning they can kill their target cells. They can move around, they can circulate, and they are often proliferating. And what happens there's really a bifurcation. If the antigen clears, so say you have an acute viral infection like a cold or the flu, these will differentiate into memory cells. And these are also they're surveilling tissues. They can move around, they can be tissue resident, but their main job is to rapidly become an effector cell again if the antigen is re-encountered. However, if the antigen persists, and this happens in, for example, a chronic viral infection like hepatitis B in humans or in cancer, you get an alternate differentiation pathway to exhaustion. These cells, this is kind of painting with a broad brush here, but they're less functional. They don't proliferate as well. They're reduced cytotoxic. And a lot of times they're sort of trapped in the tissue. They don't move around, depending on the exact type of exhausted cell we're talking about. And so my postdoctoral work, which Dan mentioned briefly, was to really narrow down on the types of exhausted T cells that are present during chronic viral infection. And so some of the highlights of that work showed that T cell exhaustion is a gradual process. There are a few different subsets that I identified. The first is was known prior to my work, a progenitor cell. These are characterized by TCF1 expression. They are generally resident in lymphoid structures like the spleen or potentially tumor-draining lymph nodes. They are mostly quiescent. They don't divide very much. They do not express cytotoxic molecules like granzyme B, and they respond to PD-1 blockade. So if you block PD-1, these cells can self-renew or they can differentiate into what we termed intermediate cells. Intermediate cells are cytotoxic. They are circulating, although they're often rare in circulation, and they're also actively dividing. And eventually, if these keep receiving antigenic stimulation, they will differentiate into terminally exhausted cells, which are characterized, for example, by expression of CD39 in a lot of human tumors. These have reduced function. They're generally tissue resident. They don't move around, and they're not dividing very often. And so the big question for our lab and many others is what processes control CD8 T cell differentiation through this pathway? Can we modify this to speed it up or to slow it down? For example, is there a way that we could cease terminal differentiation in order to keep cells in the more functional intermediate state? And so the first part of the talk I'm going to talk about today is how we used a mouse model to identify a new protein, CD7, and its role in this differentiation pathway. So from my postdoctoral work, we did some bulk RNA sequencing, essentially sequencing these three different populations. And one of the experiments we did was to compare gene expression in the terminal cells, these are sort of the most dysfunctional cells, versus the intermediate cells, which are a little more functional. And what we found is that there are many genes that are differentially expressed. There were thousands of differentially expressed genes. CD101 was the marker that we used to describe this terminal subset. But the next two highly expressed genes were one of the CD200 receptors and CD7. CD200 receptor has a known ligand, CD200. CD7 is a transmembrane protein, which I'll talk about its structure in a minute, with comparably less known about its function. It was thought to be a co-stimulatory molecule for T cells, but its function really hasn't been elucidated. So to show you exactly how much CD7 is expressed, this is actually the raw counts of CD7 from this experiment. You can see that naive T cells do express a little bit of CD7. It goes down in progenitor cells and then sort of gradually increases as T cells go down this exhaustion differentiation pathway. It's also differentially expressed between tissues. I'll show you some flow data in just a second. But CD7 is a very highly expressed molecule. For example, in the liver, if you sort out virus-specific CD8 T cells in our model of chronic infection, CD7 transcript is higher than PD-1, it's higher than TOX, it's higher than CD3, it's higher than CD8, and so on. So it's a very highly expressed gene, and so we thought it might be doing something important based on how highly it's expressed. The molecule itself looks like this. It's a relatively short protein. It's about 200 amino acids in mice. It has one immunoglobulin-like domain on the extracellular surface. It has a transmembrane helix, and then it has three phosphorylation sites in the C-terminus that have been shown to be phosphorylated through these high-throughput studies. And these are actually quite well conserved. You can see all the way back to certain species of birds that these phosphorylation sites are very well conserved. So CD7 has a very interesting expression pattern. It has a very conserved structure. But interestingly, there wasn't even a mouse anti-CD7 antibody until just about a year ago that was commercially available. So it really has remained understudied, especially in mice. And so the first thing that we did is we wanted to look at this expression pattern by flow. And so we used this chronic model of LCMV. LCMV is lymphocytic choriomeningitis virus. This is an arena virus, and its natural host is mice. And so we're looking at mice that are infected with a virus that they would be in the wild. And the way that we give it, it's actually a lifelong infection. So these mice are viremic for a lifetime after we infect them with it. And so you get this very profound T cell exhaustion as time goes on. And so we stained for CD7 on naive T cells as well as LCMV-specific cells in these tissues. And you can see again that the expression gradually increases over time. That at day 8, we really saw very little expression. By day 15, you can see some in the spleen and the liver. And then by day 45, it's even a little bit higher. And so CD7 is sort of gradually increasing over time as these mice continue to be infected. And what we also found is that CD7 is preferentially expressed on the terminally exhausted CD8 T cells. So if we look at progenitor cells, it's pretty low. If we look at intermediate cells, it's sort of mixed. And terminal cells express a lot of CD7. And this actually corresponded quite nicely to the transcriptomic data that we had generated several years before. We also wanted to look at human T cells to see if this was a similar pattern that we could find in exhausted T cells in people. And so we have this patient cohort, which I'll talk about at the end of my talk. But we had 26 patients with head and neck squamous cell carcinoma, and we essentially isolated TILs from these, tumor-infiltrating lymphocytes, and this is gated on CD8 T cells. We subdivided these into terminally exhausted cells, which are expressing PD-1 and CD39. These PD-1 positive cells that don't express CD39, which could be some sort of memory cell, it could be some sort of progenitor, as well as PD-1 negative cells. So what we found is that CD7 was actually fairly much broadly expressed in humans. There are many more cell types, CD8 T cell types that express it. But the highest expression, again, was on the terminally exhausted cells that are PD-1 positive and CD39. And so if you look at MFI, this is about maybe 2 to 3 times higher expression on exhausted cells compared to these other cell subtypes. So that was really encouraging to see that CD7 seems to be, its regulation seems to be conserved and that it's upregulated on terminally exhausted cells, both in human tumors and in our chronic exhaustion, our chronic viral model. So then obviously we want to know what does CD7 do. And what we did is we generated CD7 knockout mouse, and we did this using CRISPR-Cas9. These are whole mouse knockouts, but we crossed these to the P14 cell line. So the P14 line is a transgenic TCR line. And so what that means is every single cell in this mouse, every single T cell in this mouse contains the same T cell receptor or TCR. And these are specific for an antigen, for an epitope of LC&B, which is that viral infection model I was telling you about. So what we can do is we can isolate from these mice T cells that are either a knockout for CD7 or wild type. They contain the same TCR, and we can transfer a relatively small number of T cells into recipient mice. So in this case, we transferred about 1,000 each of CD7 knockout cells, CD7 wild type cells, and put them into a standard ballistics mouse. So you can track these by flow cytometry because of their congenic markers. And so this is what it looks like before we transfer cells. You can see it's about a 50-50 mix. And we can track these cells by flow cytometry over time as we infect these mice. And so we transferred these to mice, and then a day later, we either gave them chronic or acute infection with our LCB model, and we tracked them over time to look at their frequency, their overall numeric abundance, and their phenotype. And so this is what these data look like after you transfer. I lost my labels here, but this is essentially two different congenic markers. This unlabeled population that you see up here, these are the endogenous T cells within the mouse. This is gated on CD8 T cells, and then we can easily identify the knockout cells and the wild type cells based on their expression of these congenic markers. And so what you expect to see, which you do see here, we transferred about 1,000 cells. You'll get hundreds of thousands or millions of these cells out of these mice by day 8 because there's this huge expansion in CD8 T cell numbers after infection. And so you can see here, for example, in the lung, about 20% of the T cells were actually ones that we had transferred. And if you look at the ratio between the knockout and the wild type in these different tissues, you see that at day 8 here, it's about 1-to-1. So both CD7 wild type and CD7 knockout cells expanded just about equally after infection. But what we started to notice as time went on, this is day 15 here, you can see now there's about a 2-to-1 ratio in the liver of wild type to knockout cells, whereas the ratio in all these other tissues is about 1-to-1. This is day 45 now. You can see the ratios increased to about 3-to-1, that the wild types have an advantage in the liver. But if you look in the other tissues, again, it's not that different between wild type and knockout. And we actually have data out even farther. I'm not showing the flow here, but this seems to be progressive. Over time, CD7 knockout cells have a profound disadvantage compared to wild type cells. And this disadvantage is most striking in the liver. So what's interesting is that CD7 T cells expand normally after infection, but they have a deficit in the liver where they start to contract. But we don't see this during acute infection. So we gave these mouse the same virus. It's just a different strain, which causes viremia for about a week or so. And you see that the wild type to knockout ratio kind of hovered around 1. And so that indicates that CD7 knockout does not give any sort of disadvantage during acute infection, but it actually gives a profound disadvantage in the liver during chronic infection. And so I've talked about the differentiation pathway of CD8 T cells, and what we wanted to know is what cell types are affected in this pathway. We had an idea because we showed you that CD7 expression is highest of these terminal cells, but we wanted to actually quantify the cell types that were affected by CD7 deletion. And so we did that. This is looking in the liver. This is looking at those progenitor cells, the intermediate and the terminal. You can see there aren't very many progenitor cells in the liver. This is a non-lymphoid tissue. This is a log scale, so these are pretty low. But if we look at the progenitor, the intermediate cells, CD7 knockout did not really seem to have an effect on these cell types. But if you look at the terminally exhausted cells, you can see there's about a 60% decrease at day 45 of these terminal cells in the liver. And you may think, okay, like, is that even important, right? I told you that these terminal exhausted T cells don't have a lot of function. And so we wanted to measure that. And we measured killing capacity of these cells using a chromium release assay. And so, like I said, progenitor cells don't really have any cytotoxicity. This is where you take these P14s, you mix them 1-to-1 with a target cell expressing their cognate peptide. And you can see that, if you mix them 1-to-1, you get maybe 20-25% killing in the spleen. But actually, the liver terminal cells kill okay. They actually kill about 50% of their target cells. And this is looking a few weeks after infection. And so it may actually be important that you're losing these terminally exhausted cells, right? Because these could contribute to viral control, and that's something that I'll return to in a minute. But ultimately, we wanted to know why. Why do these terminally exhausted cells get lost after CD7 deletion? And so when you don't have any answers, as one does, you do single-cell RNA sequencing to try to generate some hypotheses. And here we had a few groups. We took naive wild type and CD7 knockout T cells. We took exhausted T cells from the liver and the spleen, both those CD7 wild type and the knockout, and did single-cell RNA sequencing. So this is a UMAP here, looking at expression of some key genes, which will tell you about the differentiation state of T cells. Naive cells express a lot of L-selectin or CD62L. So these are our naive cells. You can see that PD-1 is expressed in these two clusters. So these are the exhausted cells coming from the infected mice. TCF1 is expressed both by naive cells as well as progenitor cells. And then as I showed you by flow, CD7 is expressed by the terminal cells. So we can actually quite nicely pull out which cells are which here. So we have naive cells over here, progenitor cells here, intermediate cells in the bottom, and terminal cells at the top. Now I'm mapping this by where these cells came from. Like I said, we had 6 samples. We had wild type, naive, and knockout naive, and those pretty much clustered together. But if you look at the spleen and the liver, you see that spleen and liver cells actually clustered quite distinctly. So there are specific transcriptional programs which are associated with exhausted cells in the liver and in the spleen, and we thought that was very interesting. So we looked at the number of differentially expressed genes at these different subsets. If we look in progenitor cells, again, where CD7 is not really highly expressed, we see that there's only about 10 or so differentially expressed genes between the two samples, between the wild type and the knockout. So not that big of a difference. The same goes for the intermediate and the terminally exhausted cells in the spleen. But if we look in the liver, we actually saw hundreds of differentially expressed genes. And so what are they? Well, it's an interesting, it's sort of a grab bag. You'll see that you see effector molecules such as granzyme B and granzyme A, as well as CD7 itself is higher in the wild type. And you see some important signaling molecules such as CTLA4 and CD28, which are higher in the knockout. We also did bulk RNA sequencing on CD7 high versus low T cells from these tissues. And what we found is both in the liver and the spleen is that there were apoptotic changes, gene signatures associated with apoptosis that were higher in the CD7 high cells. We saw a similar pattern in the single-cell RNA-seq data, and so we thought that maybe CD7 is protecting these terminally exhausted cells from apoptosis, especially in the liver. And so this is the experiment we did to test that. We transferred these transgenic TCR T cells, which are wild type or CD7, about 1,000 each, put them into a standard Y6 mice. We infected it with LCMV to induce a chronic infection. Then we took out these cells 45 days later, and we stimulated them with their cognate peptide or with TMSO, which is a vehicle control here. And so what we found in the spleen, if you look at the wild type and the knockout cells, that if we measured apoptosis by active caspase-3, and you can see that in the spleen, there's really no difference, that knockout and wild type cells are about equally susceptible to apoptosis. But if we look in the liver, we found that once we stimulated with peptide, the knockout cells were about 60% more likely to undergo caspase-3 cleavage. And so this may not seem like a big difference, but if you think about it over time, over the weeks and weeks that we're looking at over the course of T-cell exhaustion, this will lead to a progressive loss of those terminally exhausted CD8 T cells. And that's why that wild type to knockout ratio kept increasing over time. So we believe these cells are more susceptible to apoptosis. And so finally, we wanted to know, does this matter for disease control, right? Does it matter that you're losing the terminally exhausted T cells? I showed you earlier that they can kill in the liver, but does that actually matter for viral control? And to answer this question, we took global CD7 knockout mouse or wild type mice littermates. So in this case, every cell in the organism is deleted for CD7. And we infected them with LCMV clone 13 and looked a few weeks later at different tissues at the viral titer. So if we looked in the serum, you can see that the viral titer was similar between the wild type and the knockout animals. The same was true for the spleen and the lung. But if we look specifically in the liver, you can see that the knockout mice had worse viral control compared to the wild type littermates by about a log. So this is about a 10-fold increase in viral titers in the liver and the knockout mouse compared to wild type mice. And so this is really interesting, right? This is interesting both for CD7's biological role as well as a broader principle, which I'll talk about in a second, which is that tissue context really matters and different signaling pathways may be important in different tissues for regulating exhausted CD8 T cells response. So this was published just a few months ago by Sean Hislop, who was a graduate student in the lab. And just to sort of recap here, what we found is that CD7 is progressively induced during CD8 T cell exhaustion, especially in the liver. That CD7 sustains exhausted T cell responses in the liver. And that if you take CD7 away, you do not get effective viral control in the liver. of these mice. And so this is a really interesting signaling pathway that we're interested in. How does it do this? What signaling is involved? What is the role of those amino acids that I showed you earlier that are phosphorylated on the cytoplasmic tail? So there's a lot of follow-up work that we're actively working on in the lab. But this also led to sort of a broader hypothesis, right? You can see that this pathway is really important in the context of the liver, but maybe it's not so important in the spleen. And so it led us to this sort of broader hypothesis is that effective T cell responses is not only about generating functional cells, making good effector cells, but also you have to sustain them. You have to keep them alive in the specific place where antigen is encountered. And so in our chronic viral model, it's actually a very broadly trophic virus. So you get virus pretty much everywhere, the spleen, the liver, all over. And it's actually a great tool for teasing out how T cell responses can be sustained. But you can imagine it's more difficult maybe when you think about cancer, which are usually restricted, at least in the non-metastatic setting, to specific tissues. It also leads to a lot of different corollary questions, such as, for example, does this hold not only between organs like liver versus spleen, but are there actually spaces within tissues where exhausted T cells could be selectively lost or maintained? Are there specific microenvironments within tissues where this is happening? Is there a specific place where the CD7 knockout cells go to die, or is it a more general phenomenon? All of these cells that I showed you in the LCV model are clonally identical. They have the same TCR. And so antigen signaling is going to be about the same, but do they experience different fates on where they reside? And is that true in tissue microenvironments? And then also, can we observe this in human tumors? As we want to go back not just to our mouse models, but can we find in human tumors what happens to clonal T cells? And of course, this question is much harder because we can't get attention to TCR T cells as an experiment. It's very hard to know antigen specificity, but we wanted to take on this challenge. And so the second half of my talk is going to be more about this work where we do this in tumors. All right. So to do this, we needed a cohort of patients. So as you know, I just moved to Baylor just a few years ago. And we developed a really strong collaboration with some head and neck cancer surgeons. So head and neck cancer globally is a cause of about 300,000 new deaths per year. It's about 50% of the new cases that are discovered each year. Most of them are squamous cell carcinoma, which can arise from multiple places within the head and neck, such as the pharynx or the larynx. And about 25% of cases are caused by infection or prior infection with HPV, which is human papillomavirus. And the rest are often associated with things like tobacco use or alcohol. Head and neck cancer is actually, in some cases, quite well infiltrated by CD8 T cells, which is usually a prognostic factor. But it's sort of a paradox because so far, immunotherapies have largely been underwhelming in the head and neck cancer setting. So it was recently approved pembrolizumab was recently approved, which is an anti-PD-1 antibody for use in locally advanced head and neck cancer, but the durable response rate, meaning that the patients who benefited for years, was about 10%. And so it's actually an open question. Why did these T cells not respond that well to immunotherapy? Why is it not that great of a prognostic factor? And are there specific tissue microenvironments factors that are driving this? And so our patient cohort was driven by collaboration with two surgeons who are both Baylor College of Medicine faculty. One works at the VA here on campus and the other at the county hospital, Ben Taub. And so we've collected dozens and dozens of samples, but we're going to talk about today about a cohort of 28 patients with head and neck squamous cell carcinoma. We got tumor tissue from all patients, and we had matched peripheral blood from all but one of them. And I want to talk a little bit about the process we use to process these samples, because I think it's really great. The people in the lab have worked really hard, and it's great to get as much as you can from these tumor tissues, which are often quite precious, right? So we get blood from these patients, and we just do a standard density gradient to get PBMCs, and we just cryopreserve them. We also get the tumors. We take a piece of this tissue and we embed it in OCT and freeze it. And so we have a long-term storage piece of tissue with its intact pathology, and it also has intact RNA, right, because it hasn't been fixed. With the rest of it, we do a density gradient to get the tumor-infiltrating lymphocytes, and we also cryopreserve those. And so this allows us to build up a cohort over time, and then we thaw all these cells, in this case at the same time. We stain them with a 30-marker flow cytometry panel, and then we use a spectral cell sorter to sort them. And so we can get this very high-dimensional flow data, which is very rich and gives us a lot of information on many cell types within the tumor. And then at the same time, we also isolated T cells, among other cell types, to do single-cell RNA sequencing and TCR sequencing. We did spatial transcriptomics with the Xenium platform on the on the tissue. And then what I'll show you later is we actually use this single cell TCR sequencing data to inform the spatial transcriptomics data to go back and look for specific TCRs in these tissues. So I'm going to very briefly show you a little bit of the flow cytometry data. Again, this is 30 color data, and you can see if we do a UMAP projection of this like you would for single cell RNA-seq, you easily pull out the CD8 T cells, CD4 T cells, B cells, and so on. And you can see some of these, such as the NK cells, are relatively similar between the PPMCs and the blood, but the T cells are very different between the tumor and the blood. And actually, the T cells are the most abundant immune infiltrate into our patient cohort. You can see the CD3 positive T cells are almost all of the immune infiltrate. There are some myeloid cells, and then B cells are in our cohort much lower. It's weird. My UMAP didn't show up here. Let me see if I can. Don't know what that happened. All right. So the first thing we did, again, is just a bunch of single cell RNA sequencing. And so we took about 250,000 T cells and we sequenced them. And we found about 11 broad phenotypic clusters. And so we found cells that are more of a naive or central memory phenotype. We found regulatory T cells, various types of memory, such as effector memory, effector cells. We found some effector cells, which I'll show you have TCRs that are very highly enriched for circulating TCRs. We found dividing cells, and we found these more terminally exhausted cells. And so we did something very simple. We did gene set enrichment analysis to look at different biological processes that are different between these different T cell phenotypes, and we saw a lot of differences. So for example, if you look at translation or protein packaging, you can see that these T cells are very differential, have very differential expression of these gene sets. You see that some of them are dividing, some of them have signatures associated with hypoxia or proteasome. So there's a lot of different biology going on in all these different T cells. And that is because human tumors have a very diverse mixture of T cells within them. And this has really become clear really over the past 5 or 10 years with some really great studies looking at, virus specific T cells that are in tumors. So we know that in tumors, sometimes you'll get naive cells, especially if you have lymphoid structures associated with those tumors. You have tumor specific T cells, which are the ones that we primarily care about, right? Because these are the ones that are actively trying to kill the tumor. These are usually exhausted within the tumor microenvironment. You may potentially have some progenitors or TPEC cells. You also have bystander cells. So you will find cells that are CMV specific or flu specific or specific for childhood vaccines, like measles. And these can be in very different phenotypic states. I'm not going to talk a lot about it, but we found that flu and CMV specific cells are very different within the tumor microenvironment. And so this can all lead to a very complex tumor microenvironment that can actually be shaped by the infections that the patient has had in the past. But one thing that you can use to tell these apart is that they will have different T cell receptors. A flu-specific T cell will have a different TCR from a tumor-specific T cell. And often these will have different TCR distributions. Tumor-specific T cells are very common in the tumor and much more rare in the blood, whereas bystander cells are often, widely distributed between both the blood and the tumor. And so for our single cell data, we actually had single cell TCR sequencing from all of this. And so this is showing you sort of those different phenotypes and outline. These are the clusters. And for each cell here, it is colored by the frequency of that cell's TCR within the tumor microenvironment. And it's a log scale. So red is very high, right? So you can see in these exhausted and dividing clusters, you will often have T cell clones that are 10% of all the T cells in that patient's tumor. And then the naive cells, for example, and the Tregs are much more diverse, and these TCR frequencies are much lower. So from all of the patient's blood that we had, we also did TCR sequencing. And so here I'm showing you the same, but now I'm coloring this information by the frequency of that particular clone within the patient's blood. And so gray means that TCR was not detected in the blood. So you can see there's a big disparity, right? If you look at exhausted T cells, you have these clones that are 10% of the T cells in the tumor, but actually, potentially not detectable at all in the blood, at least to the depth which we sequenced, which was a few 100,000 TCRs per patient. But you also see some clusters that have very high PBMC frequencies. And so you can sort of take the ratio of this and look at the tumor enrichment of these TCRs. And you can see that you do have cells that are very highly enriched in the blood, but most of them in blue here are enriched in the tumor. So this gives us a little bit of insight into what is potentially tumor specific and what is not. But the reality is it's very complicated, right? Because as I showed you, we know this differentiation pathway of exhausted T cells. And here I'm showing you one TCR that we think is tumor specific. Cells with this clone are in red, and you can see that it's in a very diverse number of states, right? So there's a little bit in this quiescent cluster. It is dividing. It is terminally exhausted. There's some effectors, which we think may be intermediate cells. And then we also have a few that look more, potentially progenitor cells. And so it's very hard to tell this in the milieu of the tumor microenvironment, because if we look here, these are T cells where we know the antigen. For example, of viral infections like CMV or EBV, you can see there's overlap, right? You have cells that look like effectors that are in the tumor that we know are specific for things like flu and EBV, and the same goes for this more memory-looking cluster here. So it's very hard if you're just looking at a T cell to understand what is this antigen specificity and what are the behaviors that it's undergoing in the tumor microenvironment. And even what we want to get into is like, where are these particular clones and what's affecting them? That's very hard to know, right? So here, what I've showed you is that these tumor-specific and bystander T cells can share phenotypic states, but their phenotype alone is not enough to infer what their fate is or their antigen specificity is. And what we really need to know is their clonal identity. And so we want to do this in a spatial manner, spatially resolved manner. And so what we need to know is, when we look at a T cell by IF or spatial transcriptomics, what in particular T cell receptor is that cell carrying? And so this led to a new project in the lab that was, we pre-printed this late last year. Hopefully it'll be out and published soon, led by a very talented postdoc in the lab, Kelly McCord. And these were the goals that we set out, right? We wanted to know in humans, how is clonal T cell function dependent on location? What are the signaling pathways that affect their function differentiation within the tumor microenvironment? And can we find where antigen-specific T cell clones are within the tumor microenvironment? Are they located in one place, or are they broadly distributed throughout the tumor? And so to do that, we needed to accomplish some technical goals, which was essentially to hopefully get single cell resolution of TCRs and the associated cells gene expression. And so to do this, we partnered with 10x Genomics and did this via in situ hybridization with the Xenium platform. And so I kind of want to show you a slide, right? Like some of the work that went into this. We do all the Xenium ourselves in our lab. We do the sectioning, we do the wet lab work, and so on. And so the Xenium slides are very expensive, as many of you may know, and you have this one capture area that you're trying to get tissue onto, right? So this was one of our first goes at it, trying to get tissue, and we got like 4 pieces. 2 of them are pretty small, and you can see that we have a lot of space that's not filled. And so we've gradually gotten better and better at putting big tissue sections and filling up most of the space, which makes these experiments more cost effective. And so Kelly in the lab, as well as a technician named Emerald, have done great jobs essentially getting this protocol working in the lab and doing a really good job. So if you haven't seen this data before, this is what it looks like, both at sort of the macro and the micro scales. This is a patient from our cohort who has a P16 positive tumor. We can actually do H&E staining in parallel with the Xenium. And so this is what that looks like. And then I'm going to zoom into this very tiny part here of this tumor. And with the kit that we used, you get 4 different stains. So you get DAPI to look at nuclei. You get boundaries. For example, you can see 1, 2, 3, and then you can see the segmented cells over here. And so this is actually performed pretty well. This is a very dense tumor microenvironment. You can see that there are lots of cells here, and it's not trivial to segment the cells, but this is actually the standard Xenium protocol. And we found that it did a pretty good job. And so what we wanted to know is, can we find T cells inside the TME, right? And so this is one cell here. I'm showing the DAPI staining. We can see that this particular cell expressed CD3 and CD8. So this is a CD8 T cell. It expresses the constant regions of the alpha and beta TCR. So this is an alpha beta T cell. It also expresses some of these molecules such as TOX and TIM3, meaning that it's probably an exhausted T cell. It expresses more granzyme A than granzyme B and no granzyme K. So this means this is probably a cytotoxic T cell. It's not a memory T cell. And then we could also look at signaling markers. So for example, this is TIGIT, which is a checkpoint molecule. And you can see that the T cell expresses it, and there are some cells around it expressing its ligand PPR. This is from an HPV positive patient, this particular cell. And so we're looking also at gene expression of the E5 gene, which comes from the HPV genome. And so you can see this particular T cell is surrounded by tumor antigen, even though it itself doesn't express any. And so if we look at an HPV negative patient, this is what that looks like. And so this was a very nice specific detection of tumor-associated transcripts. So it's one thing to look at individual cells, right? But you can't really just look at these the whole time, because, in this section, there's something like 80,000 cells or something like that. And so we needed a way to sort of analyze this data more systematically. And so we found a way. Kelly did a great job, because there are lots of cells in this data set, millions and millions of cells. So this becomes quite a large data set. But essentially, we perform a UMAP just like we do with single cell RNA sequencing. And you can see here that we found cells that clustered out quite well. So you have T cells, which are marked by CD3, CD8, for example. You have tumor cells up here, which you can see that some of them express HPV. We have myeloid cells, which express CD4, CD14. We have endothelial cells, which express in CD31. And you can do a lot of things with this. For example, you can look and see what is the composition of each cell, of each patient's tumor microenvironment. You can see most of them were dominated by tumor cells, which are shown in blue. And so we could also map these back into space, right? So going back to that cell that I showed you earlier, this was the cell that we just found visually. And then this is mapping these cluster annotations back onto these data. And you can see, for example, that this was identified as a T cell here in purple, and the rest are blue. So this is a very lonely T cell that essentially is surrounded by tumor cells here. But the cluster mapping actually did a great job of identifying these different cell types. This is a very busy slide, but we can look at, for example, what types of cells are these cells touching. So these are endothelial cells, which are in yellow. And this is essentially the proportion of its neighbors. So you can see that these very much like to touch other endothelial cells, which makes sense because they form, rings or vessels, and then there may be other stuff around them, such as fibroblasts in orange. Fibroblasts like to touch other fibroblasts because they kind of make these large structures that kind of stretch throughout the tumor, and then they touch a whole bunch of other stuff. The tumor cells are mainly in these nests, and so they touch other tumor cells. But then you see, for example, like T cells, there is some self-clustering with T cells, but they also like to touch lots of different other things. Same was true of granulocytes, and then myeloid cells also had a pretty diverse type of neighbors. But I'm going to focus in now on the T cells. We were interested in how does T cells gene expression change based on its neighbors. And this is a little bit of a complicated analysis, but it actually makes a lot of sense. What we did is we calculated for each T cell how far it was, for example, to a tumor cell, and we correlate the gene expression with the distance from a tumor cell. So if you get a negative correlation, it means it's expressing a lot of that gene near the tumor cell and kind of trailing off as it gets farther. But if it has a positive correlation, it means it's not expressing very much, but as you get farther away from those cells, it expresses more. And so if we look at the exhausted cells signatures, such as CD7, which I showed you earlier, or things like CD39 or TIM3, you can see these are very highly expressed in T cells that are near tumors, but not near normal epithelial cells. You can also see things like CD103 and CD69 are upregulated near epithelial cells, meaning maybe these are more resident memory-like. And then if you look at the stem-like markers, such as TCR7 or TCF1, we see that those are actually not very highly expressed in tumor cells, but much more highly expressed when they're near other lymphoid cells like T cells and B cells. But this is still overall a very high-level analysis. We know that, in this, for example, this UMAP of CDM T cells, we know that within this T cell cluster, because we did the single-cell RNA sequencing on the same patients, we know that there's a very diverse set of T cells within here. And so it's not very helpful to look at these broad patterns of gene expression. It's interesting, but it's not the granularity that we need. And so what we wanted to know is, can we map these very specific T cell states onto the Xenium data? And so I'm going to fly through this because the details aren't that important, but we made a machine learning model using random forest to essentially predict, based on the Xenium data, what single cell cluster it was. And it was about 80% accurate. So there are some that it got wrong, but mostly we could train these cells or this classifier pretty well to identify T cell state within Xenium. And so that led us to be able to see things like this, where we see the DAP images, we can see which ones are T cells. And then we can actually see what type it is. So the pink and the blue are exhausted. So most of these are exhausted T cells, but for example, you have a Treg up here. And so we're able to really narrow it down even further into particular T cell subtypes within the tumor microenvironment. This actually led to a lot more resolution than I thought it would. For example, looking at things like spatial clustering. So this is a statistic called Moran's I, where if it's 0, it means cells are kind of randomly distributed throughout the tumor microenvironment. And so if above 0, it means that the cells are more likely to be clustered. And then if it's less than 0, it means they're sort of dispersed. So if we pick out random cells, we get 0 here. But you see, for example, that T cells actually do have a little bit of self-clustering. T cells are a little bit more likely to be closer to other T cells than not. But this was actually differentially distributed between CD4 and CD8 T cells. And we also found that, for example, Tregs were much more likely to show that clustering near other T cells, for example, than this particular exhausted phenotype. And so it means that the behavior of T cells within the tumor microenvironment really seems to depend on their much more granular phenotype than just looking at broader, T cells as a whole. So there's been a lot of interest in recent years in interviewing niches or tertiary lymphoid structures, these aggregates of immune cells that happen within tumors, that form within tumors. And we wanted to look at this with an unbiased approach. And so we wanted to call these clusters. And so Kelly leveraged this algorithm called dbScan, which is really good at finding these oddly shaped clusters, shapes of clusters of cells that are close together, they can form these strange shapes. This often happens. So you can see, for example, this is one patient here. These are all immune cells in red and other cells in gray. And then mapping them over here, these are the CD45 positive cells, and you can see where it's calling the cluster. So for example, this cluster here was called as a cluster, whereas these more dispersed regions down here were not. And so this is sort of an unbiased way across many different tissues to identify these immune niches or lymphoid structures, whatever you want to call them, across many different tissues, right? Because we did this we have this data on dozens of patients, and we can't go through and draw circles around each one. And so looking more broadly, we can look at the T cells here and say, okay, which ones are expressing, for example, CCR7, which is a more plastic marker, more plastic T cells, versus granzyme B, which is an effector molecule. And so the positive cells are in pink. And so you can see that there are a few CCR7 positive cells, not very many, but they're largely within these clusters, whereas the granzyme B positive cells are largely outside of the clusters, right? And so this has been known through some nice work from Hayden Kissick and others, that we see this phenotype, right? That we see in clusters, which are gray, more markers expressing things, more cells expressing like TCF1, and then outside of clusters, more things like TIM3. But going back to this doesn't tell the full story because, again, like I said, clonal identity is required to interpret the spatial context. So it could be that all the flu-specific cells are inside those structures and all the tumor-specific cells are outside of them. And our analysis so far has not been able to resolve that. And so what we did to resolve this problem is to generate TCR targeting xenium probes. And so we used the single-cell RNA-seq data that we had generated from single-cell RNA sequencing, which I showed you earlier, to design probes against about the 20 most abundant TCRs from these patients. And if you're familiar with how these probes work, essentially you have transcript-specific sequences. So we designed probes against the CDR3, and these will anneal to the target RNA. They will make a circle. It will be ligated, and you get fluorescent amplification via rolling circle amplification. And so this is challenging because there's only one VDJ, one variable region per T cell receptor, versus other genes you can target with many probes, right? As many as you want. And so it's actually more of a challenge to design these, but some of them did work, and I'll show you an example here. So this is a portion of a tumor where you have multiple T cells. These seem to be mostly CD8 T cells here. These are mostly exhausted because they're expressing things like TOX and CD39. These are surrounded mostly by tumor cells. And these are two probes we designed against a paired alpha and beta TCR. And so you can see that this one T cell here, which is right here, which is exhausted, we can actually precisely identify which is the exact clone of that T cell, the alpha and the beta. And we can go back and match that to whether it's tumors. This is an example of the same clone, this particular CDR3, that's in two different places. This one is in the middle of the tumor. You can see this, the tumor cells do not express alpha SMA or remittent. T cells do. You can see this is expressing TIM3 in orange, but it's not, for example, expressing L-selectin in green. This is a cell from that same clone, which is actually more in a fibroblast-rich region, because this is alpha SMA remittent. And you can see that it's expressing a lot of L-selectin and not any TIM3. So this is a really nice illustration that we can see different TCRs, the same TCR in different places, and actually make conclusions about clonal T cell differentiation of T cells that are in different places in the tumor. So of course we wanted to validate this, right? This could be non-specific binding. And so what we did is we took tissue from multiple patients and put them on the same slide, right? So a TCR in one patient is usually not going to be present in another patient. And so we can put probes against multiple TCRs, against multiple patients, and see if we get any cross-reactivity and how much T cell enrichment there is. And so what we found, for example, this is the TCR I showed you, the paired TCR. If we take these probes from patient 9 and we put it on tissue from patient 9, we get heavy enrichment of this probe in T cells, which is what we'd expect because they're expressed by T cells. But if we take those same probes and put them against tissue from a different patient, or if we take the same patient and put different probes against it, you see that you don't see this enrichment in T cells. You get enrichment in other clusters and random things. And so what we found mostly is that probes against patient mismatched TCRs did not bind that same patient's tissue, but they did bind patient matched tissue. And so this gave us a lot of confidence that we were looking at real signal here with our TCR probes. And so I'm going to end with a few examples of what this can tell you. And so this is one patient from our cohort, and each dot here is a specific clone. So all of these blue clones are one, or these blue dots are one cell from the same clone 1 here. And so we know, for example, that all these T cells share the same antigen specificity, and we can map back what is this phenotype and where is it. And so we can find, for example, you see that in the outside of the clusters, you see there's a lot of light blue, which is exhausted, but you do see these green, which kind of look like TPEX or vector memory looking like cells. These are probably progenitors that are inside of these clusters, but largely not outside of these clusters. We can do a similar analysis looking, for example, which of these cells express granzyme B. The granzyme B negative cells are much more likely to be found inside these immune niches, these DB scan identified clusters versus outside of them. And this is quantified here. So you can see not many of these cells from this clone express CCR7, but the ones that did, or the ones that express, for example, L-selectin or CD127, were much more likely to be inside of these clusters in gray, and the cells that were much more likely that expressed TIM3 or CD39 were much more likely to be outside of the clusters. And so, to kind of sum it up, it shows you that we can identify cells expressing the same CCR, and they're therefore specific for the same antigen, at different places in the same tissue. And so, for example, we can find right here that this is a TCR clone that's in white here. We have a effector-like cell, which is granzyme B. We have one that's expressing CCR7. They're actually not that far away. This is like 100 microns away. But you can see that these are more surrounded by fibroblasts and even a myeloid cell here, whereas this is primarily surrounded by tumor cells. And so really exploring how we can use this data to understand how T cells are moving and differentiating throughout tissue. It's much harder with the human data, but now we're going back to the mouse to say, how can we apply these to mouse where we know the antigen specificities and we know the TCRs so that we can actually do a little bit more mechanism-driven research on which of these pathways that we find around these cells are important. And so to sort of sum this up, I hope I've convinced you that CD7 controls exhausted T cells persistence in the liver during chronic viral infection. And then going down to the micro level within tumors, the T cell differentiation is spatially coordinated. The same clones have different phenotypes depending on where they are. And this has really led to our labs, one of our big focuses, which is that, again, generating effective T cells is half the story, but you have to sustain them in the physical locations where they are encountering antigens. And we think this is both true at the organismal scale, that there may be different pathways that are controlling these processes within different organs, but also in the microenvironment. And so with that, I'd like to thank the people from my lab. Kelly McCord is a postdoc who just is starting a job in industry on Monday, who did all of the spatial TCR work that I showed in the second half, and she's been great. Sean Hislop is a MD PhD student in the lab who will be graduating later this year, who did all the CD7 work. Emerald did a lot with